Hello children, let us continue with the topic which we were discussing in our previous videos, that is Human Genome Project. And we had a little talk about why Human Genome Project is called as mega project, how much money was involved in it, many countries participated in making this project possible, and lot of information which was gathered from this project, how it was utilized by various industries for the betterment of the people. So these were some topics which we were continuing, and in continuation to that Human Genome Project, let us move ahead with methodologies of Human Genome Project. Now, methodologies of Human Genome Project means what was the method which was used by the scientists to crack the whole coding sequence of the DNA. That is, different types of procedure which was used by the scientists to code the sequence of the DNA. Basically, this methodology is divided into two types of sequencing. One was called as express sequence tags, and another one was called as sequence annotation. Now let us understand what does these two words means. Express sequence tag means there was an approach by the scientist. What this thought that let us first decode only that segment of the DNA which is coding for some kind of amino acid or proteins. That means that part of DNA should be coded first, which can be or which is getting transcribed into messenger RNA form. And messenger RNA, that we all know, will go for the formation of amino acid by the process of translation. So they thought that only that part of the DNA we are going to take up which is getting coded in the form of messenger RNA and later on is responsible for the formation of amino acids. This was first approach. Second approach was that let us code whole of the DNA. Let us code like whole DNA can be decoded by making the DNA into fragments. Small pieces of the DNA can be fragmented with the help of molecular scissors. There are some enzymes which can cut the DNA into small fragments and then study those fragments separately and then again rearrange them to understand the whole of the sequencing of the genome. So these approaches were used, and the more successful and the better approach was this sequence annotation, that is dividing the DNA into small fragments, was taken care of. Now in this case, what was done, I'm just reading the slide. For sequencing, the total DNA from the cell is isolated and broken down into relatively smaller fragments. Now, there are certain enzymes in the coming chapters of biotechnology, we will be discussing about them. We call them as restriction enzymes, endonucleases and exonucleases. Now, what these enzymes have the property that they can cut the DNA at a specific locations whichever we want. So, at least they can cut the DNA into small fragments. So these enzymes were first used to cut the whole DNA into small fragments. Second step, what it was taken, that this small fragments of DNA was cloned with the help of some vectors. You can read the second line. DNA is cloned by suitable vectors which can be a bacteria or an yeast. This small piece of DNA, which is now separated out, was incorporated inside a bacteria or incorporated inside the yeast, and it was allowed to, what you can say, it was allowed to multiply, it was allowed to divide. So from a single strand fragment, there many fragments were formed. So if the cloning vector which was used was inside the bacteria, that was called as bacterial artificial chromosome. If it was in the yeast, it is called as yeast artificial chromosome. One was a prokaryote, another was the eukaryote. So in that case, they were used to clone the small fragments of the DNA of the human being, which was fragmented a step ahead with the help of the restriction enzymes. Now, once they got once the bacteria multiply, the human genome will also multiply, that fragment will also multiply. So, they got huge amount of genetic code to be to decipher it out. Then what was done, that once the DNA was obtained in large number, that DNA was read and it was coded depending upon their functions and non-functions. And then because remember, in the human being we have a long chain of DNA which has many parts of the DNA has a kind of an overlapping units present. Plus, there are many parts in the DNA which are repeated units of the same sequence. Plus, there is DNA morphism, that is polymorphism, meaning a small segment of the DNA can be repeated again. Now, keeping all these properties in mind, overlapping sequences in the mind, that fragments of the DNA, after reading it, after coding it, were arranged back again, so that the whole of the human genome can be read with the help of a kind of a special automated DNA sequence program which was developed by a scientist called as Frederick Sanger. So it is called as Sanger method. So by utilizing this Sanger method, overlapping fragments of the DNA, by keeping that thing in the mind, the whole of the DNA was read. It is said that it was Sanger's method which you can say simplified the work of being coding of the DNA. And what is this actually automated DNA sequencing? It was a kind of a computer operated program in which the whole overlapping sequences of the DNA was fed, and it was the scientist Sanger who did it. So it was called as Sanger method. So by utilizing Sanger methods, the DNA which were cut into fragments, they were again joined together. Based on the overlapping regions on the DNA fragments, these sequences were arranged accordingly. That is how the whole process went. I'm moving ahead. For alignment of these sequences, a specialized computer-based programs were developed. Based on endonuclease recognition site, genetic and physical maps of the genome were constructed by collecting the information about certain repeated DNA sequences and DNA morphism. That means DNA repeated sequences plus DNA polymorphism plus overlapping sequences, they all helped in they were all present in that computer operated program which helped in bringing back the cut sequences of the DNA, and it can be read. The whole human genome can be read. Now this was actually the small methodology which was followed by the scientists for decoding the human genome. Let us have a look at the video at this stage.
Biological researchers could study only one or two genes at a time. This changed completely in 2003 with the completion of the Human Genome Project. With the entire gene sequence and advanced genetic engineering technologies at one's disposal, biological research could now be conducted on a larger scale. For example, biological researchers could now study all the transcripts in an organ, tissue, or tumor. It's also possible to study how thousands of genes and proteins work together inside our cells. HGP itself was made possible by two major methodologies of biological research: expressed sequence tags, or ESTs, or DNA sequence tags and sequence annotation. In EST or DNA sequence tag method, the focus was on identifying and isolating all genes that expressed as RNA. Actually, EST is the term for all genes that express as RNA. With EST, numerous databases of nucleotide sequences have been made available. It has facilitated the construction of the preliminary transcript map of a human genome. In the second methodology, the entire set of genome that comprised all coding and non-coding sequences was indiscriminately sequenced, and later different regions of the sequence were assigned with a function. The assigning of functions to sequences is called sequence annotation. For the purpose of sequencing, the complete DNA was extracted from a cell and divided into smaller fragments, since it is technically difficult to sequence longer fragments of DNA. These DNA fragments were then cloned in suitable hosts such as bacteria or yeasts, with the help of specialized plasmids or vectors. The vectors were called bacterial artificial chromosomes or BAC and yeast artificial chromosomes or YAC respectively. The cloning amplified each DNA sequence, which made it easier to sequence. Sequencing was then done by automated sequencers that worked on a principle developed by Frederick Sanger, who was also responsible for developing a method to sequence proteins and amino acids. As per Sanger's method, the cloned DNA sequences were arranged based on the overlapping regions present. For this, overlapping segments were generated. However, aligning this was not an easy task. As per Sanger's method, the cloned DNA sequences were arranged based on the overlapping regions present. For this, overlapping segments were generated. However, aligning this was not an easy task. Therefore, specialized computer-based programs were developed that annotated these sequences and assigned to a chromosome. Out of the 22 autosomes and 2 allosomes X and Y, chromosome 1 was the last that has been sequenced in May 2006. Did you know that chromosome 1 has the most number of genes? That is 2,968, whereas chromosome Y has the least number of genes, only 231. These challenging tasks would not have been possible if advanced genetic engineering methodologies had not been put forth by the HGP. Consequently,
Now that was the brief idea about the methodologies of the Human Genome Project, which was followed for it. Let us have a very quick go through with the salient features of the human genome, which was what was actually found out from that Human Genome Project. First point, human genome contains around 3164.7 million nucleotide bases. Now you can imagine huge amount of nucleotide bases. Nucleotide bases means adenine, guanine, cytosine and their combination. So they are having nearly this many million base pairs present in our human genome. Second, the average gene consists of around 3000 bases, and the largest known human gene, which was called as dystrophin, has around 2.4 million base pairs. Now you can imagine a gene present with us with 2.4 million base pairs present with us. So that was the largest gene which was decoded, and like that many genes are present. So remember how much the information or how much complex our DNA is. It was on an average every gene will contain 3000. So 3000 means so many genes. One gene has beyond your imagination, that is around 2.4 million bases present in it. That was called as dystrophin. Dystrophin is also involved with some kind of a disease called as dystrophin mellitus. That is it is responsible for that, where the denaturation of the muscles will start. We come down to third one. Total number of gene is estimated to be around 30,000, and 99.9% nucleotide bases are exactly same in all the people. That is why it is said that when we talk about human beings, each human being has around 99.9% same DNA. That zero point 0.1 difference on that gene sequence makes the whole story. That is why we are not similar to each other. And total genes which are present with the human, which is around 30,000. So imagine 30,000 genes, and each genes on an average comprising of 3,000 nucleotide base pairs and nucleotide bases. So you can just imagine the the whole expansion of the DNA of the human being. We move ahead. What are other silent features? The functions are unknown for over 50% of the discovered gene, and less than 2% of the genome codes for protein. Now imagine proteins, which are the main working components of the human being, for which the translation happens. Just now we have studied translation in our previous topic. That is the formation of amino acid, polypeptide chain of amino acids, which leads to protein, and the information comes from the messenger RNA, and messenger RNA is transcribed from the DNA. This DNA will have only around 2% of the whole of the DNA has the information for the coating of the protein. So you can see, you can just think that still so much of information is there in the DNA for so much of different works. It is not only the protein coding is the function of the DNA. Apart from that, there is lot of functions of the DNA, and many parts of the DNA, just now I told, they are repeated units of the nucleotides. Human genome contains large repeated sequences. That's what I was just talking about. And even after decoding the whole of the human genome, 50% of the human genome still needs more studies, needs like why it is present and what can be the uses of presence of that human genome. Repeated sequences do not have direct coding functions, but throw a light on chromosome structure, dynamics, and evolution. Now, we all agree with that presently human beings are considered to be the most evolved organism. So, we have not evolved all of a sudden. There will be many stages which we have passed on, and all those stages might have an impact on our genetic material. Probably that is why our DNA is so vast. It has still many information hidden in it which needs to be decoded. Chromosome 1 has the maximum number of genes, that is 2968, and Y chromosome has the minimum number of genes, that is 231. So chromosome 1 is the biggest, you can say the biggest chromosome in the whole of the sequence of 23 pairs when we talk about it, or 23 chromosomes when we talk about it. 1.4 million locations are there where single base DNA sequences called as SNPs or single nucleotide polymorphism occur. Now what does this mean? You can see in the diagram that from whole of the human DNA may be same. There will be only one base which will be different in many of the human beings which are related to each other. Like in one of the organism it will be adenine, other will be guanine, other will be thymine. That means there are around 1.4 million locations where single DNA sequences like SNPs occur in the human beings. They will be having just single bases which will be there, and they will be little different from the continuation of the whole of the DNA. So SNPs also make the differences in different organisms. So it's not necessary that it has to be sequence of the amino acids which will create a difference in the human beings. It can be the single base pairs, and these single bases can be millions in number, like around 1.4 million locations where the single nucleotides are present which create differences among the human beings. So this is also one of the, you can say one of the characteristic features of the human genome. What can be the future of it, applications of it, just we go through it. New approach to the biology will be radically developed when complete sequence of the human genome is properly studied. All genes in the genome and transcripts in a particular tissue, organ or tumor can be studied to understand how enormous number of genes and proteins work together. Actually, once the human genome was decoded, the miracle of the nature can be actually studied. When we say DNA is a blueprint, so blueprint is so huge and so extensive that after the human genome project was there, the scientists started understanding it out. Plus, there were many problems which are related to genetic coding. There was many problem which is related to genetic diseases also. That can be also addressed after this human genome project was completed. Moreover, the whole machinery of working of the human being, that is from the gene to the, like from the DNA to the RNA to the gene to the protein to the cell to the organ system, organism, the whole of this, you can say, a kind of a sequencing, a kind of a machinery working, cellular machinery working was properly studied, because now we came to know about the core part, that is the DNA, once it was decoded by the scientist. This is all for today's lecture. Thank you, children.